Hi and welcome to today's session. Today we'll be learning about mitosis. Mitosis is otherwise known as equational division because here the parent cell divide and form two daughter cells which will be identical both genetically and metabolically to the parent cell. So the daughter cells which are formed will be exactly the copy of parent cells. There will be no genetic variation occurring due to this mitotic division. And this mitotic division, it in case of our multicellular organisms, it is a means of regeneration, it is a means of revitalization or repair, and it occurs for the growth of that organism. While in case of while we consider the unicellular organisms, it is actually a means of asexual reproduction. So that is the case between, or that is the difference between eukaryote, multicellular and unicellular organisms in case of mitosis. And since they are identical to the parent cell, this is also known as equational division. And here actually two major events are taking place. That is, this cell has to divide and form two daughter cells. And here actually the nucleus has to divide and form two nucleus, and the division of nucleus is known as karyokinesis. And cytoplasm has also has to divide into two daughter cells, and it has to partition its content into two daughter cells. So two events are taking place: division of nucleus and division of cytoplasm. Division of nucleus is called karyokinesis and division of cytoplasm is called cytokinesis. And before the division, before the cell starts division, it has to undergo certain preparations. That is, it has to replicate its DNA, it has to produce certain amount of proteins, it has to produce certain amount of enzymes which are all needed for these processes. And also the chromatin has to undergo condensation also. Actually it may condense around 1000-fold condensation has to occur. And all these events takes place during the interphase. So the interval between the end of one mitotic cycle and the beginning of next is called interphase. The interval between the end of one mitotic cycle and the beginning of another mitotic cycle is called interphase. While the period between the end of one mitotic division and the end of next mitotic division is called cell cycle. Okay. And we know that the interphase is again divided into G1, S and G2 phase, and we have gone through all the details of that different stages. So now we will be focusing on mitosis. Mitosis, here major cellular events are occurring, and depending upon the type of events, we are classifying the mitosis into different stages. That is prophase, this is the interphase. Then the major stages of mitosis are prophase, metaphase, anaphase, telophase, and finally cytokinesis occurs. So we have to go into much details of these individual stages. First, let's move on to the prophase. In prophase, there are several events taking place. That is, first event is coiling and condensation of chromosomes, and also the polarization of chromosomes. So the first event is polarization, condensation and coiling of chromosomes. So here in the interphase time, the DNA will be seen as chromatin, less condensed form chromatin, and when the DNA is in the chromatin form, it is favorable for more genetic expression. The gene expression will be more at that time. And when it prepares to get into the division phase, that is the M phase. During the prophase, the condensation process starts and nearly 1,004-fold condensed chromosomes are formed. And then what happens? In this condensed state, the DNA cannot undergo transcription. So at this stage, the M phase, during the M phase, there will be no synthesis of RNA or any proteins. And during this time, the nucleolus also disintegrates. And the chromatin is already, or the chromosome is already duplicated. At this prophase period, the chromosome is in double-stranded form with two identical chromatids which are joined at the centromeric region. And these two sister chromatids, they are held together with the help of a protein known as cohesin. And next major event which takes place during prophase is the splitting of chromosomes and kinetochore formation. Here you can see these are the two sister chromatids, right? These two sister chromatids, they are held together by the help of a protein known as cohesin. And next event is the splitting of chromosomes. Here the sister chromatids of each chromosome, they become partially separate. The two arms become separate. Instead of being closely associated, they start separating, and the double-stranded nature, it becomes more prominent. It becomes more conspicuous. So that takes place during this stage. And also towards the end of this stage, certain proteins known as kinetochore proteins, they come and associate at this centromeric region and form a protein complex. That protein complex is known as kinetochores. So kinetochore formation occurs during prophase. And next major event is this formation of spindle fibers. That is, in these centrioles, they move to opposite poles. These centrioles move to opposite poles. Here you can see the centrioles have started the formation of spindle fibers, and before that the centrioles moves to opposite poles of the cell, and they start formation of astral spindles. These are the astral spindles, star shaped, which are not too lengthy. The spindle fibers which are formed in the two opposite poles are called astral rays. Now the centriole and the astral rays, they are called as astrosphere. And after that, this centrosome region, it transforms to a elongated body, a tube-like elongated body, and that tube-like elongated body is called centrodesmus. Okay, and certain other fibers, mitotic fibers, you can see this arise from this centrosome. And that this is the another type of event that is taking place. And next phase is the pro-metaphase. During this stage, the nucleolus starts dissolving. Here you can see there is no nucleolus during the prophase, right? Interphase, we have a clear and intact nucleus, and you can see clearly the difference between the two stages. Here it is, the genetic material DNA is in the chromatin stage. Chromatin-like organization is seen, thin thread-like, which condenses to form this condensed chromosome structure. And here there is no intact nucleus. So during pro-metaphase, the nucleolus and both nuclear membrane both becomes dissolution. They start disintegration. And nuclear envelope disintegrates to the cytoplasm as elements of rough endoplasmic reticulum from which it was initially formed. And that details we have gone through during the topics we were learning during the cell organelles. With the disintegration of the nuclear envelope, the central part of the cell becomes a fluid zone, which is condensed chromosomes, which randomly move about in disorder. You can see here. Here there is no central region. Here only a fluid-filled region is seen, and the chromosomes move around simply. Next phase is metaphase. In metaphase, what happens is, in this prophase, the chromosome, they start movement, they start poleward movement, or they start centrifugal movement. That is centrifugal in the sense around the periphery to or towards the periphery of the nucleus. Okay, normally the chromosomes are oriented or arranged inside the nucleus. Here the as they form this condensed structure, they start moving towards the periphery of the nucleus, and then nuclear membrane dissolves, nucleolus dissolves, and they are seen in the central region of the cell as this randomly moving bodies. And during the metaphase, what happens, they start moving and they come and align in the central plane. The movement of chromosomes here is towards the center of the cell, and this centripetal movement is called metachinesis, or we can call it as concretion. The chromosome, they get attached to the chromosomal spindles. You can see the spindles coming and attaching to this kinetochores. So here the spindle fibers get attached to the center of the chromosomes, and they align along the central plane or the equatorial plane of the cell. This is called orientation. And this central plate is called metaphase plate. The chromosomes, they get aligned in the metaphase plate, and this enables the separation segregation process of the chromatids towards the opposite poles. And during anaphase, what happens, the separation of the sister centromere starts and the segregation of sister chromatids occurs. You can see half of this chromosome breaks along the central line, and half of the chromosomes are pulled to the opposite poles, as you can see here. So here the sister chromatids, they move to the opposite poles in a uniform velocity, and this movement is attained, or this is achieved with the help of this organization and disorganization of these spindle fibers. And this separation is actually thought because of the repulsion force between them, which is created by an interaction between the kinetochore and the spindle fibers attached to it. And in this way, the cohesins, which are the proteins which made the sister chromatids to closely associate with each other, which that breaks down. So the cohesin breaks down, and the breakdown of cohesin is attained by the enzyme separin. So separin comes and it breaks the cohesin. So the sister chromatids, which were closely aligned, now the attachment is withdrawn and they move towards the opposite poles. So that is the process which happens during anaphase. And in telophase, this is the end of the poleward migration of the chromosome, and the chromosome are tightly coiled, and then they remain in two groups which are on the opposite poles of a cell. So the cellular events are concerned, as far as it is concerned, the telophase is the reverse of prophase. Here the chromosomes which are now condensed, it starts to decondense. So the decondensation of chromosome occurs, and nuclear envelope reappears, nucleolus reappears, mitotic spindles dissolve, and the cytoplasmic cleavage starts. So that are the major events which takes place during the telophase. So telophase is actually here the reverse events of prophase occurs. And now They form a reticulum in each pole of the parent cell, and after that nuclear envelope is formed around each reticulum, and which is probably derived from the rough endoplasmic reticulum. And nucleolar region organizes, it again comes, nucleolus again reappears. That type of events all occur during the telophase. And next is, now the nucleus has been formed. Now the cytoplasm has to divide. So the division of cytoplasm is called cytokinesis. Cytokinesis, it is the division of cytoplasm which leads to the formation of two daughter cells. So that occurs, and here all this process occurs in a coordinated manner, or it is in a regulated manner. Any deviation from this regulation may lead to the formation of neoplastic cell. All these divisions are taking place in a controlled manner. If that control is lost, that cell may become neoplastic. Neoplastic, another term is cancerous. So in that case, there is no control over the division, and it randomly divides to form multiple number of cells, which forms the tumors. So that is the case which is occurring in cancerous conditions. So all these cellular events are taking place under controlled conditions. There is a regulation in all these division processes. So that is all about mitosis. Thank you.